The thunder. Quran chapter 13, verse 4 highlights how fruits and crops, despite being irrigated with the same water, can vary in taste and quality, influenced by carbon in the soil. Carbon, as organic matter, impacts nutrient availability, water retention, and microbial activity, all crucial for flavor development. Quran chapter 13, verse 12 speaks of divine signs like thunder and lightning, which contribute to the nitrogen cycle by producing nitrogen compounds that fertilize the earth and support life, underscoring the interconnectedness of natural forces. Both carbon and nitrogen, with atomic numbers 6 and 7, are essential for the fertility of the earth and the development of life and flavor. And on the earth there are different neighboring tracts, gardens of grapevines, various crops, palm trees, some stemming from the same root, others standing alone. They are all irrigated with the same water, yet we make some taste better than others. Surely in this are signs for those who understand. Arad 13 verse 4. The carbon content in soil has a significant and multifaceted impact on the taste of fruit. This is primarily through its role in soil organic matter and the soil microbiome, both of which profoundly influence nutrient availability, water retention, and overall plant health. The foundational element here is soil organic carbon, SOC, which forms the very essence of healthy, vibrant soil. It's not just inert material, rather, it represents the living, breathing component of the soil system, derived from the decomposition of countless generations of plant and animal residues, along with the biomass of the microscopic life that teems within. This rich organic matter serves as an extraordinary nutrient reservoir, a biological bank that meticulously stores essential plant nutrients. Unlike synthetic fertilizers that offer a rapid, often fleeting, burst of nutrients, the decomposition of organic matter ensures a gradual, sustained release of these vital elements, nitrogen, phosphorus, potassium, and a spectrum of micronutrients like boron and zinc. This steady, balanced nutrient supply is paramount because it directly fuels the plant's metabolic machinery. It's the engine for photosynthesis, enabling the plant to produce the very sugars that define a fruit's sweetness. It supports the synthesis of organic acids, which are critical for the pleasant tartness and overall balance that makes a fruit truly palatable. Furthermore, this consistent nutrient stream allows for the intricate creation of volatile organic compounds, VOCs, the complex, aromatic molecules that give each fruit its unique fragrance and contribute profoundly to its perceived flavor. Without this stable nutrient delivery, fruits can often taste bland, exhibit underdeveloped sweetness, or lack their characteristic aromatic complexity. Beyond its role as a nutrient custodian, soil organic carbon profoundly enhances the soil's water-holding capacity. Imagine it as a giant, incredibly efficient sponge woven throughout the soil structure. This remarkable ability to absorb and retain water is a game-changer for fruit quality. It acts as a crucial buffer, protecting fruit development from the detrimental effects of both drought stress and excessive moisture. During dry spells, this stored water is slowly released to the plant's roots, preventing the plant from experiencing the severe stress that can result in smaller, tougher fruits with concentrated, sometimes unpleasant, flavors. Conversely, in periods of heavy rain, the improved soil structure fostered by organic carbon allows for better drainage, preventing waterlogging that can suffocate roots and lead to disease, again compromising fruit quality. A consistent, well-regulated water supply ensures that fruit cells expand properly and sugars are efficiently transported, resulting in juicy, plump fruits with a consistent, delightful taste. Crucially, soil organic carbon is the lifeblood of the soil microbiome. It provides the primary food source and the intricate architecture for the vast, diverse communities of bacteria, fungi, archaea, and other microorganisms that call the soil home. These microscopic allies are not merely passive residents, they are active partners in the plant's growth and taste development. Many microbes tirelessly work to solubilize nutrients, converting insoluble forms of elements like phosphorus into plant-available compounds. Others engage in the remarkable process of nitrogen fixation, pulling inert nitrogen gas from the atmosphere and transforming it into usable forms for plants. Beyond basic nutrient acquisition, there's growing evidence that the soil microbiome can directly influence a plant's secondary metabolism, subtly guiding the production of compounds that are not strictly for survival but are key to defense, communication, and, most relevantly, flavor. This might involve the production of phytohormones that fine-tune ripening, or even direct precursors to those very VOCs that delight our senses. A thriving, diverse microbial community also bolsters overall plant health and resilience, making the plant less susceptible to pests and diseases. A less stressed plant can then dedicate more energy and resources to developing highly flavorful fruits. Finally, soil organic carbon significantly improves soil structure and aeration. By binding tiny soil particles into stable aggregates, it creates a porous environment. These pores allow for optimal air circulation, providing the oxygen essential for healthy root respiration and efficient nutrient uptake. They also ensure proper water infiltration and drainage, preventing root damage from compaction or anaerobic conditions. Healthy, well-aerated roots are fundamental to a plant's ability to absorb everything it needs from the soil, directly impacting the intricate biochemical processes that culminate in the superior sweetness, acidity, and complex aromas of a truly delicious fruit. Based on the comprehensive review, the sweetness of grape berries is fundamentally a story of carbon accumulation. Essentially, all the carbon that forms the sweet-tasting sugars is imported from the leaves. Through photosynthesis, leaves fix atmospheric carbon dioxide into sucrose, which is then transported via the phloem to the developing berry. The berry's sweetness is not determined by the leaf's ability to produce sugar but by the berry's own highly regulated capacity to unload, process, and store this imported carbon. The fate of this carbon changes dramatically at a key developmental stage called veraison, which marks the onset of ripening. Before veraison, most imported carbon is used for growth or channeled into metabolic pathways that produce organic acids, primarily malic acid, making the berry tart. At veraison, a major shift occurs. The berry switches its phloem unloading strategy and its internal metabolism. Now, the imported sucrose is systematically broken down into the hexose sugars glucose and fructose, which are then actively transported for storage. This final step of storing sugars is managed by specialized transport proteins. Families of transporters like SUTs, HTs, and most importantly, TMTs and sweets, act as gatekeepers, moving the hexose sugars across the plasma membrane and into the vacuole of the berry's flesh cells. Here, they accumulate to extremely high concentrations. Therefore, a grape's sweetness is the direct result of this efficient, multi-stage process of carbon import, metabolic conversion, and active transport into storage within the berry.